Hello, so here's your snow day edition of microbiology, and it's chapter 5, microbial metabolism. Now, why do you have to know about the metabolism of microbes? It's because, well, when they infect something, they start to use you as a dinner plate, and we have to talk about some of the processes that they go through and the ramifications of infection. And growing them in a laboratory, we need to know their metabolism. So here are the three things. You have to discuss and compare the processes common to life. You would be very surprised to find out that microbes carry out the same sort of important metabolism that we do. We need to talk about enzymes, pathways, how enzymes are inhibited. Hopefully that's a review. And discuss the role of enzymes in metabolism, how we can control those pathways for our own uses, what we can learn from them. Those are the objectives. All right, so you remember metabolism is the sum of all the chemical reactions in a cell. The ultimate goal of metabolism is to reproduce the organism. That's easy. So here are some statements that go along with metabolism. And we have to agree that every cell requires nutrients. The energy to break down those nutrients, either catabolism or anabolism, can come from light or breaking down things like sugars and fats. I don't like this phrase because we don't store ATP. Organisms do not store ATP, but ATP is made in order to provide energy for other things. So cells catabolize nutrients. That means they break them down to form other molecules. So the best example of this is to break down proteins so that you have a pool of amino acids to make more proteins. Precursor metabolites and energy end up in anabolic reactions to make new macromolecules. And then cells grow by assembling these macromolecules. Don't forget your macromolecules are proteins, lipids, carbohydrates, and nucleic acids. Right? Let's see what you remember about this. Catabolism is the breaking down of metabolites. Anabolism is the assembly of them. Catabolism usually releases energy. Anabolism requires energy. And that energy is usually released and stored with the help of enzymes. Enzymes are very specific for the reaction they carry out. And just as a review, we're going to have to talk a little bit about oxidation and reduction reactions. So I like the Leo-Szesger, L-E-O-G-E-R. Lose electrons oxidize, gain electrons reduced. Some people say oil rigs, oxidize, lose electrons, or reduction is gaining electrons, whichever one you like. So enzymes, they're the things that lower the transition energy for a reaction to happen. So without an enzyme, you might need lots of energy, lots of heat, lots of time, but enzymes make that all go away by lowering the reaction transition energy needed for that to happen, activation energy. This does not work. Okay. Here are the types of enzymes, and you need to know these. So hydrolases break things apart. A really good example of that is breaking a peptide bond with water. It's hydrolysis. Isomerase, if you remember your chemistry, iso means a difference. So isomerase is just rearrange atoms within a molecule. Ligase, we're going to talk about a lot when we talk about DNA synthesis or replication, when we talk about DNA engineering. Polymerases are the enzyme that string nucleic acids together. Lyases split chemicals into smaller parts without using water. So hydrolysis is with water, lyase is without water. Oxidoreductase is transferring electrons or hydrogen atoms from one molecule to another. A good example of that might be NADH, NADPH. And transferase is moving a whole functional group from one molecule to another. Those are very special enzymes. We'll see those in glycolysis. All right, here's an enzyme, and these are all vocabulary words associated with enzymes, most of which are not on your vocabulary sheet. But you should remember that enzymes have active sites, that enzymes have places for direct inhibition and allosteric inhibition, which I'll show you in a minute. Enzymes have cofactors. Those cofactors can be organic, like NAD has vitamin B in it, or inorganic, like magnesium ions or iron. So you should remember your cofactors. Here are some cofactors. Magnesium is inorganic. Organic, remember, NAD, that's the long name of NAD. It basically carries two electrons and a hydrogen. The coenzyme is vitamin B. NADPH, that's in plants. FAD, that's in the Krebs cycle. Carries hydrogens, carries two hydrogens. We're not going to talk about this one. Coenzyme A, we're going to need to talk about when we talk about glycolysis. We're not going to talk about peroxidol phosphate or thiamine pyrophosphate. Okay, here's an enzymatic activity diagram. So we have fructose 1,6-bisphosphate. This is a step in glycolysis. It enters the enzyme. The enzyme rearranges that, causes stress on the internal bonds, and breaks that apart into glyceraldehyde 3-phosphate and dihydroxyacetone, which goes in to become another chemical in glycolysis. Remember, the enzymes are specific for just one molecule. So this enzyme won't work on any other molecule except fructose 1,6-bisphosphate. The reason that's good for us and microbes is because if we have a microbe and it makes a nice tidy little enzyme that could do work for us in an industrial application, like detergents for washing your clothes or making a cheese, well, we just take that one enzyme and we know it's only going to impact this one particular chemical. All right, so enzymes, you know that enzymes don't work very well in high temperatures or low pH. But if you get the perfect temperature and the perfect pH, they work at an optimal rate. Enzymes depend on substrate concentrations and enzyme concentration. If you don't have enough enzyme, you're going to have a slow turnover of substrates. If you have much more substrate than you have enzyme, you're also going to have a slow turnover. Inhibitors of enzymes can be toxins, say from mushrooms. They can be pharmaceutical products like drugs. Inhibitors can block an enzyme's active site, or they can allosterically regulate an enzyme. We're going to talk about that a little. Inhibitors do not denature enzymes. Remember, enzymes are proteins, and if we subject them to high temperature or low pH, they melt. That tertiary and secondary structure melts. And if you're not, if you're trying to remember about this primary structure, secondary, tertiary, review chapter 2. That's going to be very important to understanding this chapter. Okay, so here's a graph of an enzyme's activity at certain temperatures. And you can see that at about 36 degrees is the optimal temperature for this enzyme. So this is where you can convert the most substrate. This would be about human body temperature. If we were to put ourselves in the refrigerator, our enzymes don't work very well, do they? We get cold, which is going to happen Thursday night. But if you have a bacteria, let's say Salmonella, that contaminates chicken. If you have Salmonella and you store your chicken in the refrigerator, is that Salmonella going to effectively be able to spoil your chicken in a short amount of time? Not really, because you're going to slow its metabolism down to maybe right around here. A refrigerator should be, a freezer is 4 degrees Celsius. A refrigerator should be about 20. So we're going to take that enzyme activity right down to here, and your chicken will stay, with a relatively low dose of Salmonella for probably 3 days. All right, pH. The optimal pH for this is about 7. Again, that's what human bodies pH like to be around. So if you've got a bacteria that also likes to be at that pH, and you preserve it with vitamin C or citric acid, then you're going to slow down that bacterial metabolism. It won't be able to grow and divide. And this is just a graph talking about enzyme activity in relation to how much. Okay, this should be a review, but we'll go through it. How can enzymes be inhibited? The best way to inhibit them is with competitive inhibition. This means that the substrate where it binds in the active site has something else that can bind there. So a competitive inhibitor binds to the active site right here, binds to the active site so that the substrate cannot bind, therefore competitively inhibits it. So if you do that, most things that we will encounter of that nature will be pharmaceutical drugs to stop bacterial infections. When we get to that chapter, you'll see a number of drugs that are competitive inhibitors of enzymes that bacteria have. Mushrooms have toxins that are competitive inhibitors. I think it's mostly what we're going to be concerned with is antibiotics. All right. Now, there are other kinds of inhibitors called allosteric inhibitors. These are chemicals that work at a site other than the active site. So allosteric means other than the active site. And if you can look at this diagram, once the allosteric inhibitor binds, it changes the shape of the active site so that the substrate can no longer bind. This is temporary, whereas competitive inhibition can be permanent. Allosteric inhibition is temporary. And this is how you would regulate enzymes so that they are not always on and not always off. When we talk about the lac operon, if you remember that from biology, the trip operon, those have allosteric inhibitions. All right, so that's allosteric inhibition. This talks about activators. You can actually have enzymes that are kind of off, and then this would be the trip operon, where an allosteric has activation abilities for the enzyme. So you can shut it off with an allosteric inhibitor, you can activate it with an allosteric activator. This is all under the heading of how you would control enzymes. Okay, remember this? I'm glad I'm recording this so I can't hear the moans. So this is glycolysis. This is the Krebs cycle. This is the electron transport chain. Bacteria have the same pathways we have for energy production. Everything on the planet, whether it's an algae or a protist or a bacteria, all have glycolysis as a pathway. Some have the Krebs cycle as a pathway. Some have the electron transport chain. Some have the ability to ferment. Even we have the ability to ferment in our muscles. What do I want from you in this particular class? I don't want every single enzyme that you will have to memorize in biochemistry. I want the big items that come out of glycolysis. So that's NADH, ATP, pyruvic acid. What happens to the pyruvic acid afterwards is what you'll have to keep track of depending on the kind of bacteria. So if it's something like a yeast, which is a eukaryote, or trying to think of a bacteria that ferments, it would go into the fermentation pathway where we could have carbon dioxide to rise bread, or we could have ethanol to produce wine and beer, and we could have other byproducts of fermentation, or the pyruvic acid can go into the acetyl-CoA shuttle and then into the Krebs cycle. Depends on the bacterial pathway we're studying or interested in. The Krebs cycle, you remember, goes around and around with a bunch of enzymes, but the big products of the Krebs cycle are ATP, NADH, and FADH. Now, do you remember that these things, the NADH and the ATP, all go into that electron transport chain? In eukaryotes, it's in mitochondria. In bacteria, it's on their cellular membrane. It's on the inside of their membrane. And this makes sense if you remember that you probably talked about the endosymbiotic theory in biology. It's also covered in your book, if you don't remember, where the evolutionary ancestors of mitochondria and chloroplasts were free-living bacteria. So mitochondria used to be bacteria, chloroplasts used to be bacteria. Mitochondria perform electron transport on their membranes, therefore bacteria perform electron transport on their membranes. Very cool stuff. So out of the electron transport chain, the big take-home message is lots and lots and lots of ATP because of ATP synthase. Remember that? You get about 38 ATP. Now let's talk about aerobic versus anaerobic. So aerobic bacteria are going to use glucose, they're going to use the Krebs cycle, and they're going to use the electron transport chain because oxygen is the final acceptor for an aerobic bacteria. An anaerobic bacteria, the one that you're trying to grow in lab, Clostridia, is anaerobic, doesn't want oxygen. So it will go through the glycolysis pathway, it will go through the Krebs cycle, and it will go through the electron transport chain, but it won't use oxygen as a final electron acceptor. It can use CH3, it can use nitrite, it can use sulfite. Those are final electron acceptors for anaerobic bacteria. And the difference is that they'll make between 34 and 36 ATP, and aerobic bacteria will make between 36 and 38 ATP. So they get a little boost for using oxygen. Let's point out some other key things. Glycolysis does not require oxygen. So if I ask you the question of which pathway does not require oxygen, you would say glycolysis. And I would say anaerobic bacteria do not use glycolysis because they don't like oxygen. And you would say true or false? False. Everything uses the glycolysis pathway. It does not require oxygen. Nowhere do you see oxygen in or out. So take-home messages, what are the products of glycolysis? NADH, ADP, pyruvic acid. What are the result of the Krebs cycle? NADH, FADH, ATP. That all goes into the electron transport chain, where the end product is either water or nitrates or sulfates or methane. Okay, this is just some background on glycolysis in case you don't remember it. This is a great little animation. I encourage you to watch it. It only takes a couple minutes, and it's on the regular PowerPoint. All right. This is a close-up of the Krebs cycle, just reinforcing what you get out of the Krebs cycle. This is the electron transport chain. Everything in red is important. Think about how this transport chain works. If you were to somehow damage the membrane of a bacteria, could you still have the production of a proton gradient? Would you still have a viable bacteria if you disrupt the membrane and then disrupt the electron transport chain? No. So a target of antibiotics is often the membrane so that they cannot carry out electron transport and they can't produce energy. So think about not just remembering these steps and how they work, but the application of that in terms of mitochondria, or in terms of bacteria, sorry. Okay. This is just a fancy way to remind you how the electrons travel on the electron transport chain. More importantly, I put in an animation for you to view that. This is a great diagram. Don't get dizzy. This shows a membrane. This would be the same in mitochondria. It would be the same in bacteria. So it shows all the 4 cytochromes. Remember, those are cytochromes. Those are massive proteins that pass the electrons down. As the electrons are passed from cytochrome to cytochrome, hydrogens are evolved. The hydrogens turn into a proton gradient. And do you remember what happens to the proton gradient? That's used to turn ATP synthase. That gigantic protein complex actually rotates like a motor, and the proton gradient makes it happen. Remember we talked about protons and flagellums? This is the same thing. Proton gradient turns this. It squeezes ADP and a phosphate into ATP. How many ATP for aerobic bacteria? How many ATP for anaerobic bacteria? Okay. All right. So this is what I was talking about before, about applying what you learn. So if you remove oxygen, what happens to the electron transport chain? If you add cyanide, which disrupts the membrane, what happens? This is called uncoupling. If you do anything to disrupt the chain, it's called uncoupling. So think about those things. All right. Chemiosmosis is basically the electron transport chain. That's what it's called, chemiosmosis. This is from the tutorial. I just really need to drill it into you, the importance of that transfer of electrons and the building of the proton gradient, because that's how they're going to make all that ATP. If you don't have that ability, you're going to be a bacteria that only makes 4 ATP, and you're just not going to grow very quickly if you're only making 4 ATP. So if you have a bacteria that's been forced into anaerobic conditions, it's facultative aerobe, you can force it into anaerobic conditions, into a fermentation pathway. How much energy is it going to produce? Not a lot. If you give it oxygen, then it's going to choose to go down the other way and make a lot of ATP. But mostly we like to use the fermentation pathway because those end products are the ones we usually want, like ethanol. Okay, these gives you where most of the ATP is produced, and of course the NADH and the FAD are used to generate that hydrogen proton chain. Okay, here's some products that are the results of the fermentation pathway. You'll be studying this in lab. We're going to take a few bacteria, including your unknown bacteria. You're going to put it into 4 different kinds of sugars, like glucose, lactose, maltose, and you're going to find out if that bacteria can ferment those sugars. And what happens when it ferments them is it turns yellow because it produces acid. When you make fermentation, you produce carbon dioxide, which you can just see in the tube, and you make acid, which turns this pH indicator yellow. This is sauerkraut. This is better living through disgusting bacterial reactions. And sourdough bread is yeast, and that's all about the carbon dioxide. Okay, don't pay any attention to that. That was a different book, but I love the chart. So glucose to pyruvic acid. These are different bacteria. This is a yeast. This is bacteria. We use Clostridium in industrial processes to make acetone and isopropanols made by bacteria, like your nail polish remover, your rubbing alcohol. It's all made by bacteria now. Of course, beer and wine. Lactic acid to produce cheese, to produce vinegars. This is all done with bacteria. This is a Swiss cheese. That's CO2 and that kind of biting flavor of Swiss cheese you get from this bacteria. Propanea bacterium, aspergillus, which is a fungus, lactobacillus, streptococcus, Saccharomyces, and this is Clostridium. So, on an exam, if I were to ask you for 2 byproducts of bacteria, you should be able to come up with some of these and the bacteria that produce them. So pick your favorites. All right, so this is really telling you those electron acceptors for anaerobic bacteria. So anaerobic bacteria can use, I don't know why it does that, nitrate, sulfite, and carbonate, and they can produce things like methane. Those are the ones that make swamps smell bad. Sulfites, sulfates, nitrates. This is what you get happening in aquariums when you have a good bacterial cycle. And this tells you how many ATP. Notice that fermentation does not produce a whole lot of ATP. Okay. What happens to fat molecules? Bacteria, some bacteria do have the ability to digest lipids. If you leave wheat too long or some fat-containing things too long, they will go bad. They don't sour. They get like they smell really bad. It's because bacteria have started to degrade the lipids. Can't think of the word right now. But it's a glycerol backbone, remember, with fatty acid chains, which are just carbons and hydrogens. So these bacteria have the ability, using the enzyme lipase, to break the fatty acids from that glycerol backbone. So you have free fatty acids. We're going to do an experiment in lab where a media will change color based on the pH change created by these free fatty acids. So it is incumbent upon you to remember that the monomers of proteins are amino acids, the monomers of lipids are fatty acids, the monomers of DNA are nucleic acids, and the monomers of carbohydrates are glucose, fructose, things like that. Bacteria can use 2 methods to digest fatty acid molecules, lipids. They can use the lipase pathway, or they can use a pathway that uses NAD and FADH. It's called beta oxidation. This one uses water. We're mostly going to be concerned with the water one. Okay. Rancid, that's the word when lipids go bad. Something becomes rancid. Wheats can become rancid. Things with high fatty content become acid. So here's a polypeptide, a chain of proteins. Proteases, we're going to do an experiment in lab where we're going to use like skim milk. And we're going to look at a bacteria's ability to produce proteases and break down proteins into amino acids. It's called deamination, and they enter the Krebs cycle. Fatty acids can enter the Krebs cycle too. So lipases for fatty acids, proteases for proteins. Okay. In the chapter, I think there's 2 pages on photosynthesis, and we're just not going to go there. But here's an animation to help you review photosynthesis. We're not going to do a lot with algae, but understand that they do use photosynthesis, and if you need a little bit of review for that, see the animation. Okay. This is in here. You don't have to memorize this or anything, but I just want you to start making connections between the products that come out of glycolysis or where they can enter glycolysis. So amino acids can enter glycolysis and be used to make enzymes. Glycerol can enter glycolysis. Peptidoglycan for the cell wall comes out of glycolysis, and fructose 6-phosphate. I think how we would use this is if I were to say, where is the acetyl-CoA used? You would be able to say the Krebs cycle, and acetyl-CoA is important for fatty acid or lipid breakdown. If I said, where is pyruvic acid, which you know is one of the outcomes of glycolysis, that's important in the production of amino acids for enzymes. The big stuff. All right, so here's a close-up of something that happens as an outcome of glycolysis, and this is called gluconeogenesis. This is new glucose production. If a bacteria happens to store a lot of polymers of carbohydrates, they have the ability to break them down. So do eukaryotes. So gluconeogenesis. You're going to have things come into this cycle like acetyl-CoA. Instead of going into the membrane and entering the Krebs cycle, acetyl-CoA can also be used as a shuttle to put these things in to make starch, to make glycogen, and to make peptidoglycan for the cell wall. So if I had a question like, what is a product in bacteria of gluconeogenesis, you'd be able to say peptidoglycan. Good. All right. Amino acids can enter the gluconeogenesis cycle. Glycerol can enter it. Fatty acids. The Calvin cycle, you should remember, just happens in plants and photosynthetic organisms, but they also can do gluconeogenesis. Right. Okay. I blacked out this because I don't want you to know all the mess in here, but I think it's in your book. So Calvin-Benson cycle is particular to fats, glycerol, fatty acids. I just want to, you know what, let's not even worry about this. We have the exam coming in, and I just don't want to even bother you with that. So you're free. That's a free pass. Okay. Work smarter, not harder. So cells only synthesize enzymes when the substrate is available. If a bacteria is growing in a glucose and lactose broth, it preferentially wants to digest glucose, not lactose. So there's no enzymes present to break down lactose. They use the most efficient energy choice. Once the metabolite is gone, once the glucose is gone from the broth, then the bacteria would turn on the ability to break down lactose. Okay. I like the next 2 charts. They seem a little busy, but I just wanted to give you a different way to focus on some of the things that can happen. So let's see if we can break this down a little bit. Where does a bacteria generate its nucleic acids? So if you follow it back, it comes out of glycolysis, and you notice that nucleic acids can both go in and come out. Proteins come out of the end of glycolysis with the help of acetyl-CoA. So just about, here's fatty acids. The big picture is that bacteria, just like us, have the same metabolic pathways. They have the same pathways to produce lipids, to break down lipids. That's anabolized lipids, catabolized lipids, proteins, amino acids. And the way that we can observe these things is by using media in a laboratory. oftentimes we want to be able to distinguish between bacteria based on its metabolic characteristics, which is how you will identify an unknown bacteria in a couple weeks if we ever have class. So you'll be given some unknown bacteria and you'll have to carry out these series of tests by which you will be able to characterize its metabolic characteristics. This bacteria produces proteases, which breaks down amino acids. This particular bacteria cannot undergo fermentation of glucose, but it does ferment lactose. You'll have all these clues as to what this bacteria is by the enzymes it produces. And because it produces those enzymes, or does not produce those enzymes, means that it has the genes for that or does not have the genes for that. Because now it's easy for us to take a particular gene and put it into another bacteria if that's what we need. All right, this is just a good diagram. This is another one that all of this happens, all of these metabolic tricks go to make sure that a bacteria can reproduce. Now, having said that, if we can control and limit those pathways through competitive inhibition, allosteric inhibition, we can prevent them from dividing, or we can slow and control that growth. And the next chapter is all about what bacteria need to grow. So first we had to look at the kinds of pathways and metabolic processes they have, and then we can look at what they need to grow. We briefly touched on temperature and pH, and the next chapter we'll talk a lot about growth restriction, temperature restriction, pH restriction, and you did that in lab this week. You have bacteria growing at different temperatures. You're going to see which they prefer. You're going to see different pHs and which they prefer. Putting it all together, okay? Any questions, bring them to class. If you have questions after class and before the exam, just email me. I will be around on Friday to talk to anybody, okay? I know it's a big push because of the snow days, but don't freak out about it. We'll get through it together and make, any kind of adjustments we need to make, okay? All right. Thanks. Have a good night.